Well, hello and welcome to our first online lab for Cell and Molecular for this fall semester. It's going to be about SDS gel electrophoresis and affinity purification of antibodies. And it's going to use results from last semester's student results for the experiment that they did. Then unfortunately, you just get to analyze results and not do it yourself. Okay, so we use rabbit serum as our starting material. Serum doesn't contain blood cells, but it contains many, many proteins. Okay, albumin is the most abundant of these, but other important ones include immunoglobulins, specifically IgG antibodies, as well as many other proteins that are involved in things like transport, blood coagulation, and many more. There are thousands of different proteins in serum, actually. We didn't kill a rabbit, though. We bought our serum. Okay, so we're going to use affinity chromatography to purify the IgG. By purifying, I mean separating it from the rest of the proteins in the serum. And we have to know something about the structure of these proteins in order to think about the purification. Okay, for one thing, it's a tetramer, meaning there's four protein subunits in there, two each of the heavy chain, which is the larger one, and two each of the light chain, which is the smaller one. So the whole molecule has a molecular weight of about 150 kilodaltons, and it's made of the two copies of the 50 and two copies of the 25. And we will see those on our gel. Okay, so here's a representation of an IgG. You'll often see this particular representation because it makes it pretty clear what's going on with this molecule. The light green parts are the light chain. There's two of them, and the darker green is the heavy chain. And we can see that they're associated physically with one another, and you can see the disulfide bonds that are connecting them, some of the disulfides. So these are the molecules we're going to try to purify, and we will. Okay, so protein purification in general, affinity in particular. The idea is you're going to physically separate the protein of interest from what may be a very complicated mixture, complex mixture of proteins in any cell or biological fluid. So the way this is accomplished many times is exploiting the differences between the protein that you're interested in versus the rest. And we know, of course, that proteins differ in many ways: size, shape, charge, and ability to bind other substances. So we're going to use affinity purification, which relies on specific binding of IgG to a particular protein called protein A. Okay, so what we're representing here is a mixture of proteins that contain your protein of interest. And we're going to pour it over in a matrix in a column such that the matrix contains a substance to which the protein of interest can bind, those are indicated by the little orange things on the round beads. So the idea is that most of the proteins will ignore the matrix with the bound ligand because they don't bind to it. But the protein of interest does bind, and therefore the other proteins can be washed through by just putting more and more buffer through. The proteins of interest stay stuck. And then by changing the conditions, we can then elute or take away the protein that we want. In our case, it's going to be IgG. Okay. So I think I probably just said this. Most of the proteins do not bind the ligand, wash through. The target protein does bind, and therefore it is separated from the rest. But then we have to treat it in such a way to release it from the column so that we can then capture it in a separate test tube that we can then use for whatever purpose we want. Okay, so IgG binding to protein A agarose is extremely popular, very important technique. These are proteins that are naturally produced by some species of bacteria, and they bind IgG very tightly. So that's going to allow us to separate the IgG from the rest of the proteins in the serum. And it's important to recognize that it's the constant region of the IgG, the stem part, that actually will bind. And these protein A and or G beads can be purchased. So what those are little tiny bits of essentially agarose or separose to which the protein A is covalently bound. This slide represents what's happening when you're binding protein A, G agarose, attached to a bead, to antibodies in solution. The antibody, when it's encountered by the protein A or G agarose, binds to it by the stem region and is retained by the column after extensive washing to remove other proteins. The binding is very firm, but the immunoglobulin can be released by changing the conditions such that it can then later be eluted from the column, releasing itself from the protein A or G. This slide shows purification of IgG using affinity chromatography, in this case using protein G bound to agarose beads in a column. And what's represented here is the little pink triangles are your IgG, and they're in a complex mixture, namely serum, and that when it's poured through the column, most of the proteins do not interact with the protein G, and they wash right through. So then rinsing the column a little bit cleans those out, leaving us with the protein IgG, protein G complexes still present on the column. Then changing the conditions, which is to say high pH in this instance, allows release of the IgG from the column, in which case when we collect that material into test tubes, we can then later analyze the antibodies that we have acquired. And then the column, of course, can be regenerated and reused again. So fractions are collected and then analyzed. Okay, there's going to be some videos later that you should watch. Okay, so we used SDS polyacrylamide gel electrophoresis then to observe whether our purification was successful. If you think about the previous slide, we simply took our material that we thought was going to be IgG and put it in a test tube. We don't know it's IgG. We need to have some other way of telling that that's what we're really looking at. So in the second part of this, we're going to be talking about SDS polyacrylamide gel electrophoresis in general, but also as it pertains to analyzing the IgG that we've collected from our little column. Okay, so this is part 2, our SDS polyacrylamide gel electrophoresis. Okay, the idea is to observe the presence of the proteins in the serum and to observe whether or not the purification of IgG was successful. Now, this technique has gazillions of applications besides this. Okay, so here's the whole procedure in a nutshell, which is from your book, and I'll come back to this again later. I'm putting it here so you can sort of visualize what a gel looks like. We've got the sample being loaded into this gel apparatus. They're showing us where the gel is, and we're going to apply a current across the gel. And then we're going to have to, before we run the gel, apply certain treatments to the proteins to make them behave themselves so that we can determine something about their size. Okay, so SDS polyacrylamide gel electrophoresis. Electrophoresis itself involves migration of proteins in response to an electric field. Polyacrylamide gel is made of cross-linked acrylamide polymers. It's kind of a gooey, slightly sticky substance, and the proteins are going to travel through it when we apply the current to it. Okay, at our lab and labs, we buy our gels pre-made, but many people can make their own. Okay, so important part about this is SDS gel electrophoresis, SDS page, is done using denatured proteins, and I need to take a minute or so to explain that. Okay, so the principle is the electrophoresis, we're going to put a current across the gel. The gel on the bottom is a plus, and that will force the proteins through the gel. The acrylamide exerts a drag on the proteins, allowing the smaller ones to move more quickly than the larger ones. Okay, so the idea is we're trying to separate individual proteins by their size, with the smallest ones going fastest and the larger ones proportionally slower. So what we're trying to see is bands of proteins where the placement of the band tells us how big the protein is. Now this won't work if we use native proteins because they differ so much in size and charge and everything. So we need to denature our proteins and make sure that they're all negatively charged in a uniform way in order for this to work out. Right, we've got proteins non-denatured have different charges according to their amino acids, sequence, pH. So they're not all negatively charged. They might not migrate at all. So we certainly need to make sure they get a negative charge so they'll migrate towards the plus pole. Okay, another issue with native proteins. Some of them have only one subunit, but many others exist as protein complexes, which have more than one subunit. That's, you'll hear, as quaternary structure. And there are many non-covalent interactions that keep those things together. And proteins also have very different shapes resulting from secondary and tertiary structure, so that they're not uniform in any respect in terms of their size, their charge, their shape, their subunit constitution, or anything else. and that's what we want it to be like in order to be able to separate them. In addition, many proteins contain disulfide bonds, in which pairs of cysteines are linked to each other, and we need to break those in order to separate protein molecules from each other. So the treatment of proteins prior to the SDS polyacrylamide gel itself is in order to ensure that the proteins have uniform negative charge, that they exist only as monomers, and that all the disulfide bonds have been removed. Okay, so to denature the proteins, we treat them with a combination of heat, SDS, sodium dodecyl sulfate, which is a negatively charged detergent. We also include a reducing agent, such as dithiothietol or beta-mercaptoethanol, and those are going to break the disulfide bonds. So as a result of these treatments, the proteins attain uniform charge and shape, and they will migrate according to their sizes in the polyacrylamide gel. Now, proteins don't have any color to them, so if you just load them on a gel and turn the power on, you won't see anything. So the samples are always mixed with a loading dye so we can see whether the gel is run far enough. The loading dye very often contains glycerol in order to keep the sample in the well to make it heavier than the buffer. Here's what SDS looks like, and here's what a protein looks like before SDS. This is supposed to represent a native protein where it has some charges, but some plus and some negative. It also has hydrophobic areas, which are being sequestered from the solvent. But after SDS treatment, the SDS coats the protein uniformly along its length, basically obliterating the native charge of the protein and giving it a uniform negative charge and uniform linear shape. Here's another representation where the sulfates are the negative charge, and they're just kind of lined up, coating the protein on the outside and giving it the negative charge that we need for it to have. Okay, here's our disulfides. Again, here's what a cysteine looks like. If we have the two of them connected by disulfide bonds, we need to break those, and that's what the dithiothietol or beta-mercaptoethanol will do to separate those subunits from each other, and also to get rid of any crosslinks within the molecule that will interfere with its proper physical conformation. So here's a more detailed structure of IgG to see what we're up against. We saw the disulfides that connect the heavy and light chains, but there's also a bunch of disulfides connecting various regions within the molecule. And we've got lots and lots of secondary and tertiary structure, as well as a quaternary structure, which is the binding of the different subunits to each other. So the SDS and dithiothietol and heat will take care of all that for us in order to make them run properly. All right. This is what I just said. So the denaturation treatment will allow them to run correctly according to their subunits, which, reminding you, are 50 kilodaltons and 25 kilodaltons. Here's a picture of our apparatus again. We can imagine this gel being loaded. We've talked about the SDS, we've talked about the disulfides, and now we need to look at the actual running of the gel using pictures from Biorad and other people to show you what it would really look like if you were going to do it. Okay, I think I said this. Oh yeah, one thing important, standard proteins of known sizes are generally placed in at least one of the well. Because if we have a bunch of proteins we're running and we don't know how big they are, we need to have some kind of a standard to compare them to. So normally what one does is acquire or purchase mixtures of proteins whose sizes are already known and run those along with our unknown proteins in order to be able to calculate the actual molecular weights of our unknown, right? So in our lab, we use pre-stained protein standards so we could actually see them during electrophoresis and know that the gel was running correctly. Okay, so here's a picture of a gel. It's as we purchase it. It comes in a plastic bag with inside a plastic pouch to keep it from drying out. Okay, so you take the gel out of the plastic pouch. The green thing is the comb, and when you take it out, it forms indentations in the gel, and that's where the protein samples can go. Okay, it also comes with handy-dandy markings on it with the gel lanes numbered, so you can actually see what you're doing when you put your samples onto the gel. The gel is very thin, and it's wedged between two pieces of plastic plates, called plates, And we have to separate those after the electrophoresis to get the gel out so that we can stain it or treat it further, whatever that may be. Here's a picture of a gel rig, the kind we would use, or apparatus for loading and running the gel. And you could look at that on the video. And here is when you've torn apart the plastic plates, and the gel itself is kind of floppy and gooey and easily ripped if you're not too careful. So this is what it actually looks like after you get it out of the gel apparatus. Here's another picture of the gel after electrophoresis. Now this one was not run. If it had been run, there would have been a blue line from the loading die. This is just one that I opened and sacrificed for the purpose of illustration. Right, so you have to stain the gel or do something else to them. We use Kamasi Blue in lab, and the bands appear along the length of the gel according to how far the particular protein migrated. Okay, and so here's a picture of a gel that was run by a class last year, and the lanes on the left are the serum that was successively diluted to give you actual bands versus giant blobs on the gel. The known proteins are in the second lane from the right, and the lane on the right contains our, well, let's say partially purified IgG. You can see the heavy and the light chains on there. Okay, so there's a couple of videos to watch about this. They're all very short. And for our lab itself, we will analyze the results from previous student experiments, along with answering a few questions about acrylamide gel in general.